In 1998, three American scientists won the Nobel Prize in Physiology or Medicine for a discovery that surprised much of the medical world: that a simple, unstable gas made of just one nitrogen atom and one oxygen atom, something most people would associate with air pollution rather than human biology, turns out to be one of the most important signaling molecules in the entire human body. That molecule is nitric oxide, and in the decades since that Nobel Prize was awarded, research into its role in human health, and particularly in how we age, has expanded into one of the more scientifically rich and practically useful areas of aging research available today. Nitric oxide production naturally declines as we get older, and this decline has been linked in substantial research to many of the changes we associate with aging, from reduced cardiovascular function to declining exercise capacity to changes in cognitive performance. Understanding what nitric oxide actually does, why its production falls with age, and what the evidence says genuinely helps support healthy levels of it, gives us a window into one of the more interesting and actionable areas of the aging science that's emerged over the past 2 decades. To understand why nitric oxide matters so much for healthy aging, it helps to understand what it actually does inside the body, because its function turns out to be remarkably central to how our cardiovascular and metabolic systems operate on a daily basis. Nitric oxide is produced primarily by the cells lining the inside of our blood vessels, called endothelial cells, through an enzyme called nitric oxide synthase, which converts an amino acid called arginine into nitric oxide as one of its byproducts. Once produced, nitric oxide diffuses into the smooth muscle layer of blood vessel walls, where it triggers relaxation of that muscle, causing the blood vessel to widen, a process called vasodilation. This single function, causing blood vessels to relax and widen, turns out to have enormous downstream consequences throughout the body, since it directly influences blood pressure, blood flow to every organ and tissue, and the delivery of oxygen and nutrients to cells throughout the body, all of which depend on blood being able to reach them efficiently. Beyond this vascular function, research has identified nitric oxide as playing significant roles in several other processes relevant to healthy aging. It's involved in regulating how effectively platelets, the blood cells responsible for clotting, behave, with adequate nitric oxide, helping prevent the kind of excessive clotting that contributes to heart attacks and strokes. It plays a role in immune function, where certain immune cells produce nitric oxide as part of their mechanism for fighting off pathogens. And more recent research has identified nitric oxide as playing a meaningful role in mitochondrial function, the process by which our cells generate energy, along with emerging research suggesting connections to cognitive function through its effects on blood flow to the brain, and potentially through more direct signaling roles within brain tissue itself. Now let's talk about why nitric oxide production declines with age, because understanding this mechanism is essential context for understanding why the interventions we're about to discuss actually work the way research suggests they do. Research has identified several distinct but interconnected reasons for this age-related decline. The enzyme responsible for producing nitric oxide, endothelial nitric oxide synthase, has been found in multiple studies to become less active with age, producing measurably less nitric oxide from the same amount of available arginine compared to younger tissue. A change researchers believe relates partly to accumulated oxidative stress affecting the enzyme's function over decades of cellular activity. Separately, research has found that oxidative stress, an imbalance between damaging free radical molecules and the body's antioxidant defenses that becomes more pronounced with age, directly degrades existing nitric oxide after it's produced, essentially neutralizing it before it can exert its beneficial effects on blood vessels, meaning the age-related decline in nitric oxide availability reflects both reduced production and increased destruction happening simultaneously. Chronic low-grade inflammation, which as research across many areas of aging science has established tends to increase with age, has also been found to interfere with nitric oxide signaling pathways, adding yet another layer to why this decline accelerates as the years accumulate. The clinical consequences of this decline, as measured across numerous studies examining vascular function in aging populations, include measurably reduced blood vessel flexibility and responsiveness, elevated blood pressure, reduced exercise capacity due to less efficient blood flow to working muscles, and researchers increasingly believe some contribution to the cognitive changes associated with aging, given how dependent brain function is on adequate responsive blood flow. With this mechanism established, let's move into what the research actually supports as effective ways to help support healthy nitric oxide production and availability as we age, since this is genuinely one of the more evidence-rich areas of practical aging science currently available. The first and perhaps most extensively researched category involves dietary nitrates, compounds found abundantly in certain vegetables, particularly beets, leafy greens like spinach and arugula, and to a lesser extent, celery and other vegetables. This might seem like an unusual starting point, since dietary nitrate itself is not nitric oxide. But research over the past 2 decades has revealed a genuinely elegant biological pathway connecting the two. When dietary nitrate is consumed, a portion of it is converted by bacteria living on the surface of the tongue into nitrite, which is then further converted, particularly in the acidic environment of the stomach and elsewhere in the body, into nitric oxide, representing an entirely separate production pathway from the enzyme-based pathway discussed earlier. One that research has found remains relatively well preserved with age, even as the enzyme-based pathway declines, making it a particularly valuable target for supporting nitric oxide levels, specifically in older adults. Multiple randomized controlled trials have examined the effects of dietary nitrate, often delivered through concentrated beetroot juice in research settings for precise dosing, and have found measurable improvements in blood pressure, in the efficiency of blood flow, and in exercise performance following supplementation, with some of the more striking findings involving improved exercise capacity and reduced oxygen cost during physical activity, essentially meaning the same exercise intensity required less oxygen and felt easier following dietary nitrate intake. A finding replicated across numerous studies involving both athletic and older, more sedentary populations. Research specifically examining older adults has found that this population, given their already reduced enzyme-based nitric oxide production discussed earlier, may actually derive proportionally greater benefit from dietary nitrate intake compared to younger populations whose enzyme-based production remains more robust. An encouraging finding suggesting this particular dietary strategy may become increasingly valuable, not less, as we age. The second major evidence-based strategy is regular aerobic exercise, and the research connecting exercise to nitric oxide function represents one of the more consistent and mechanistically well-understood findings in this entire field. Exercise increases blood flow and creates a mechanical force against the walls of blood vessels called shear stress. And research has found that this shear stress directly stimulates the enzyme responsible for nitric oxide production, essentially meaning that exercise doesn't just temporarily increase nitric oxide during the activity itself, but appears to produce lasting improvements in the underlying enzymatic machinery responsible for ongoing production over time. Multiple studies examining regular exercise training programs, generally spanning several weeks to months, have found measurable improvements in endothelial function, a standard research measure of how well blood vessels respond and dilate in response to stimuli, closely tied to nitric oxide availability. And importantly, this research has found these improvements occur even in older adults, and even in those with existing cardiovascular risk factors, suggesting the vascular benefits of exercise on nitric oxide function remain genuinely accessible throughout the aging process, rather than being limited to younger, already healthy populations. Research comparing different exercise intensities has generally found that both moderate continuous exercise and higher intensity interval-based exercise produce meaningful improvements in this area, with some studies suggesting interval-based approaches may produce somewhat greater improvement in endothelial function per unit of training time. Though the more important finding across this body of research is simply that consistent aerobic activity, of essentially any reasonable intensity and type, produces measurable and meaningful benefit for this specific aspect of vascular aging. The third strategy involves antioxidant-rich foods and their relationship to the oxidative stress pathway discussed earlier as one of the key drivers of age-related nitric oxide decline. Since research has established that oxidative stress directly degrades nitric oxide after it's produced, foods and dietary patterns that reduce overall oxidative stress represent a genuinely distinct and complementary pathway for supporting nitric oxide availability compared to the direct production pathways discussed so far. Research on polyphenol-rich foods, including berries, Dark chocolate with high cocoa content, green tea, and pomegranate has found associations with improved endothelial function and markers of nitric oxide availability, with some clinical trials specifically measuring these outcomes, following controlled intake of these foods and finding measurable improvement compared to control groups. Flavonols found concentrated in cocoa have received particular research attention, with several studies finding that regular intake of cocoa products high in these specific compounds, distinct from most commercial chocolate products which lose much of this content during processing, produces measurable improvements in blood vessel function. Improvements researchers have connected mechanistically to both direct antioxidant protection of existing nitric oxide and some evidence suggesting these compounds may also support the enzyme-based production pathway directly. The fourth strategy involves adequate intake of specific nutrients that serve as direct building blocks or essential cofactors within the nitric oxide production pathway, beyond the dietary nitrate pathway already discussed. The amino acid arginine, mentioned earlier as the direct substrate the nitric oxide synthase enzyme converts into nitric oxide, has been studied extensively, including in supplement form, though research findings here have been somewhat more mixed than the dietary nitrate research, with some studies finding meaningful benefit for blood pressure and vascular function, particularly in populations with existing cardiovascular risk factors, while other studies have found more modest effects. A pattern researchers believe relates partly to how efficiently supplemented arginine is actually absorbed and utilized compared to arginine obtained through whole food sources, and partly to individual variation in how much the enzyme-based pathway specifically, as opposed to the dietary nitrate pathway, is limiting nitric oxide production in a given person. Vitamin C and vitamin E, both established antioxidants, have shown research support for helping protect nitric oxide from the oxidative degradation discussed earlier, with some studies finding that adequate intake of these vitamins is associated with better preserved endothelial function with age, reinforcing the broader theme that supporting nitric oxide involves addressing both production and preservation simultaneously. The fifth strategy, less discussed in popular conversation about this topic but genuinely well supported in the research, involves avoiding or minimizing factors that actively impair nitric oxide function, since protecting existing production and availability matters just as much as actively boosting it. Smoking has been found in extensive research to significantly impair endothelial function and reduce nitric oxide bioavailability through multiple mechanisms, including direct oxidative damage and impaired enzyme function, with research finding that this impairment begins to reverse, though not always completely, following smoking cessation, reinforcing smoking cessation as one of the more significant interventions available for this specific aspect of vascular health, alongside its many other well-established benefits. Certain mouthwashes, interestingly, have been found in research to interfere with the oral bacteria responsible for the first step of the dietary nitrate to nitric oxide conversion pathway discussed earlier, with studies finding that regular use of antibacterial mouthwash measurably reduces this conversion process and correspondingly reduces blood pressure benefits normally associated with dietary nitrate intake. A genuinely surprising and practically relevant finding that has led some researchers studying this area to specifically caution against regular antibacterial mouthwash use in people, particularly focused on optimizing this pathway, though this should be weighed against mouthwash's legitimate oral health benefits and discussed with a dental professional rather than treated as a simple universal recommendation. Chronic excessive sugar intake and the insulin resistance it can contribute to over time have also been linked in research to impaired nitric oxide signaling, adding another connection between the broader metabolic health topics we've covered in other contexts and this specific vascular aging pathway. Sleep also deserves mention within this discussion, since research examining sleep quality and duration has found measurable associations with endothelial function and nitric oxide-related markers, with studies finding that both insufficient sleep and poor sleep quality, including conditions like untreated sleep apnea, which causes repeated drops in blood oxygen levels during the night, are associated with impaired vascular function through pathways that research suggests involve both increased oxidative stress and disrupted normal nitric oxide production patterns, reinforcing yet again how interconnected these different pillars of healthy aging research consistently turn out to be. Bringing these strategies together, what emerges from the substantial body of research now available on nitric oxide and aging is a genuinely coherent and actionable picture. Regularly consuming nitrate-rich vegetables like beets and leafy greens, maintaining consistent aerobic exercise, incorporating antioxidant and polyphenol-rich foods, ensuring adequate intake of supporting nutrients through a generally well-balanced diet, avoiding smoking, being thoughtful about factors like excessive sugar intake, and interestingly, habitual antibacterial mouthwash use, and prioritizing good quality sleep, together represent an evidence-backed approach to supporting one of the more fundamental and far-reaching signaling systems in the entire human body. What makes this area of research particularly compelling is how directly it connects to outcomes people care about deeply as they age: cardiovascular health, exercise capacity, energy levels, and increasingly cognitive function, all trace back to a molecule so simple in its chemical structure that it consists of just two atoms, yet so central to human physiology that its discovery reshaped our understanding of how the body regulates itself, and continues decades later to offer some of the more genuinely practical and well-supported guidance available in the entire field of healthy aging science. It's worth understanding how researchers actually measure nitric oxide-related function in living people, since this helps explain how confident the field can be in these findings. Because nitric oxide itself is extremely short-lived, breaking down within seconds of being produced, researchers rarely measure the molecule directly, and instead rely on downstream markers, most commonly a technique called flow-mediated dilation, which measures how much a blood vessel, typically in the arm, widens in response to a brief period of restricted blood flow followed by release, a response that depends heavily on nitric oxide signaling and has become something of a standard research tool for assessing vascular health non-invasively. This measure has been validated extensively against actual cardiovascular outcomes, meaning studies have found that people with poorer flow-mediated dilation scores do, in fact, go on to experience higher rates of cardiovascular events over time, which is part of why researchers trust this as a meaningful proxy when studying interventions like the ones discussed throughout this conversation, even without measuring nitric oxide molecules themselves directly. It's also worth acknowledging individual variation in this research, since not everyone responds identically to the same intervention. Genetic differences in the nitric oxide synthase enzyme itself, along with baseline differences in oxidative stress levels, existing cardiovascular health, and even gut bacterial composition, which as research has found plays a role in that oral to nitrate conversion pathway discussed earlier, all appear to influence how strongly a given person responds to strategies like dietary nitrate intake or exercise training. This is less a limitation of the research than a reflection of the genuine biological complexity involved, and it reinforces why the strongest recommendations emerging from this field tend to involve combining multiple complementary strategies, dietary, activity-based, and protective, rather than relying on any single intervention in isolation, since doing so gives the greatest likelihood of meaningfully supporting this system, regardless of where any individual's particular limiting factor happens